Good morning, students. Myself, Major Komal Katyal, Department of Zoology, GK Gwani Government College, Peenmal. Today, I will deliver a lecture on antibodies, and this is my sixth lecture in the series of lecture on immunology. आज हम बात करेंगे प्रतिरक्षी के बारे में, जो मेरा छठा व्याख्यान है प्रतिरक्षी तंत्र से संबंधित. Now, in the beginning, we will discuss about what are antibodies. Antibodies are assembled out of protein chains, and there are many different chains that the immune system assembles in different ways to make different antibodies. If we look at the diagram shown in this slide, you can see the antibodies has two clear-cut chain. One, the heavy chain here shown with the light red and dark red color, and the orangish colored light chain. So a antibody has a heavy chain and a light chain, and both has a constant region which is light colored and a variable region which is dark colored, and a yellow colored antibody is attached at the variable region. Apart from it, if we look at what type of cell antibodies are produced by, antibodies are produced by plasma cells. I've already told you regarding the B lymphocytes that in the B lymphocytes certain cells which produce antibodies are known as antibody producing cells, AFC, or the plasma cells. These antibodies are essential part of adaptive or acquired immunity because the antigen antibody reaction is specific. So the antibodies specifically bind a unique antigenic epitope, also called as antigenic determinant. As I've told you during my lecture on antigen, that antigen has epitope or antigenic determinant, and likewise the antibodies has paratopes. So they have the antigen binding site which are known as paratopes, and they are the members of class of proteins known as immunoglobulins. Now antibodies belongs to that class of gamma globulin fraction of serum proteins. They are basically Y or T-shaped polypeptides, and we have already seen that they have two identical heavy and two identical light chains, and they have a constant which is blue colored here and a variable which is orange colored region, and they have the antigen binding site. So there are 5 different kind of antibodies. जो प्रतिरक्षी हैं, प्रतिरक्षी सामान्यतः क्या होते हैं, Y या T आकृति के पॉलीपेप्टाइड हैं जिसमें 2 हैवी और 2 लाइट चेन श्रृंखला होती हैं पॉलीपेप्टाइड की, और 5 प्रकार के प्रतिरक्षी पाए जाते हैं: IgG, IgM, IgA, IgD and IgE. Now each heavy chain has a constant and variable region which I told you earlier on. Each light chain has also a constant and variable region, and they have a FAB, that is fragment antigen binding, and they have FC, the fragment constant. You can see in the diagram the CH1, the constant heavy 1, 2, 3, as well as variable heavy shown with the VH. Likewise, the constant light is labeled as CL and variable light labeled as VL. So this is about the constant and the variable region. Now antibodies can also be divided into 2 regions based on their functions. The tip of Y consisting of 1 constant and 1 variable region, each from the heavy and the light chain of the antibody, contains the region that binds the antigen as shown here in the diagram. It is for this reason that we call this region as FAB, fragment antigen binding region. The base of the Y, composed of two heavy chains only, plays a role in modulating immune cell activity, and this region is called FC, that is fragment crystallizable. Now, the Fab is a variable region of the antibody present at the tip, binds the antigen, and it is specificity of the antigen determine this region. The region FC is present only in the constant region at the base, and this region can bind to cell receptors, complement proteins, as well as to other immune molecules, thereby initiating different physiological effects including opsonization, cell lysis, and degranulation. Now about the antibodies and how they function. The antibodies can act as receptors. They have a soluble Ag that is secreted in the blood and tissue, and they have a membrane bound Ag which is found on the surface of B cell, also known as B cell receptor, the BCR, as you can see here in the diagram. The function of antibody as receptor. The antibodies can attach to a B cell and serve to recognize the foreign antigen. The second one is antibodies as effectors. The free antibodies which are present in our blood sera can bind to antigen which tags the antigen for the immune system to attack and destroy. So here the antibodies are functioning as effectors. Now let us discuss about the general function of antibodies. The antibodies released into bloodstream will directly attack the invading agent. It means they can perform their function by two ways. This is the first way. It includes the agglutination, which we can easily see during the blood transfusion, the precipitation, neutralization, lysis, opsonization, tissue fixation, and selective transport, that is transferred from mother's maternal tissue to the developing fetus through placenta. And second, the antibodies may activate the complement system, and this complement system include opsonization and phagocytosis, the lysis of the foreign microbes, chemotaxis, and activation of mast cells which are responsible for inflammation and producing certain allergic conditions. Now we will discuss about the various type of antibodies. As I've told you earlier on, there are 5 different type of antibodies. Number 1 is IgG, immunoglobulin G. It is approximately 70 to 75% of total immunoglobulin present in the blood serum. It is secreted in high quantities, especially in the secondary exposure. What is secondary exposure? Secondary exposure, it means when that particular antigen attacks our body for second time. This is the secondary exposure. They are the only antibodies which can cross the placenta. It means the IgG is the only antibody which pass from maternal tissue to the developing fetus. केवल यही प्रकार की प्रतिरक्षी क्या होती है, मादा से भ्रूण के अंदर अपरा द्वारा जा सकती है. The major function or application of this antibody includes the neutralization of microbes and toxin produced by such microbes. Second, it opsonizes the antigen for phagocytosis. Third, it is responsible for activating the complement system. and it is present in the newborn baby, so it protects the newborn. And you can see the structure. It has the light as well as heavy chain and disulfide bond plus the joining J chain. Whatever we have discussed earlier on as a generalized structure is the structure of immunoglobulin G. Now, this antibody has four-fold rise or fall indicates on active infection, and secondly, a single positive sample indicates the past exposure to that particular antigen. The second type, that is IgM or immunoglobulin M, it is secreted initially during the primary infection. It is the first antibody which is released whenever an antigen attacks our body for the first time. It is a pentamer, and due to its large molecular size, it cannot cross the placenta. It has a short half-life of 5 days only. The major function includes it is secreted during the primary exposure, and then it activates the complement system used as a marker of recent infection. So whenever there is concentration of this antibody, it means the infection is recent. It is presence in the newborn means infection. The single positive sample in serum or cerebrospinal fluid indicate recent or active infection of a particular disease, and it is used only to detect the early infection or the early phase of the infection. The third type, that is immunoglobulin A or alpha, the IgA, it is a dimer basically with secretory component in the lumen of GI tract or the gastrointestinal tract and in the respiratory tract. It is basically secretory present in the colostrum, the first milk of the mother after the birth of a child is known as colostrum, and this IgA is present in the colostrum means providing the immunity to the newborn baby. It neutralizes the microbes and toxin produced by the microbes. It is present in the serodiagnostics of TB or tuberculosis and synthetically respiratory virus test. If this immunoglobulin test positive, it means you have certain respiratory problem or infection. The fourth one, the immunoglobulin D or IgD, is a monomeric with a short half-life of 2 to 3 days, limited only to the bloodstream, and the major function includes it is present on the surface of B lymphocyte, function as a membrane receptor because it is present at the surface only. Its role is still not clear, but had definitely role in antigen stimulating lymphocyte differentiation. Structure more or less similar with the immunoglobulin G. Now the last one, that is the fifth type of antibodies, that is immunoglobulin E, IgE. It mediates the first type of hypersensitivity. It does not have a hinge if we look at the structure. It is heat labile. It means it is found only in traces. Very few plasma cell can synthesize this, and it has also a short half-life of 2 to 3 days. It is associated with anaphylaxis, plays a role in immunity to helminthic parasite, and associated with allergic diseases, especially in the developed country where asthma and hay fever occurs. So serodiagnosis of infectious and non-infectious allergy can determine the presence of this immunoglobulin, as in the case of aspergillosis or parasitic disease. The sequential of IgM and IgG include: first there is secretion of IgM, and then there is secretion of IgG after the secretion of IgM. m, and this is what we can see through the graphic. This finishes our lecture on